Okay. Can everyone hear me? Okay, good. Okay, so I am a student that is a master's student working out of Elizabeth Clare's lab at York University. And my master's research looks at using a novel method of airborne eDNA to, in this case, survey bat roosts. So eDNA, for those of you who don't know, stands for environmental DNA. And it's fragments of DNA that we get from the environment that isn't directly taken from an organism. So this can be bits of hair, skin cells, saliva, or even extracellular strands of DNA. We've detected it in the soil, in water, and even some unconventional methods like honey or in the guts of vertebrates that feed on things like blood, so mosquitoes. Airborne eDNA, however, is relatively new. So up until now, at least for surveying the terrestrial environment, has been primarily theoretical. Most practical work has looked at pathogen and bacterial communities, with a little bit looking at plants and fungi communities, using these dust traps here. And the way these work is they passively sample the wind, essentially, and settle particulate matter out of the air, and then you take that particulate matter and extract eDNA from it as you would a soil sample. In 2021, my supervisor published a proof of concept paper that was performed in a very controlled environment that showed that we could in fact get airborne eDNA of terrestrial vertebrates out of the air and use it to identify what they were. Since then, there's been a handful of studies that have expanded on that and looked at less controlled environments. And again, we were able to find that we could still detect those vertebrates with pretty high accuracy. So what I wanted to do for my research was use that active sampling method to see if we could use airborne eDNA to detect bat biodiversity. So I used these three samplers here. For this experiment that I'm talking about today, it's only the large ones. But we had 3 different prototypes that we took down to our field site, and there's an example of them being deployed behind a bat line in our field lab. So I wanted to specifically look at roost surveys. This is a potentially non-invasive, semi-remote way of surveying roosts, or at least that's what we thought it would be. The previous studies that had looked at this work suggested that cavities, tree cavities, caves, burrows would be an ideal use of this method. So we wanted to test that out under truly natural conditions to see if we could detect wild bat populations. And we deployed our samplers in 4 different roost type categories in the Archaeological Laminae Reserve, the Kakabish Mayan ruins, and the Indian Creek area of the Orange Walk district in Belize. So the first roost type category was the natural caves. An example here, this is called the Schoolhouse Cave, and I'll be coming back to talk about this a little bit later. The second kind of group was man-made tunnels made into Mayan ruins by looters and archaeologists. The third was other man-made structures, and this consisted of an old cistern and then an attic. And finally, of course, tree roosts. So here's some of our tree roosts and then our other natural cave. For some of the actually all the tree roosts, we couldn't actually enter the tree roosts either because the entrances were too high or too small. And then for this cave here and then one of our tunnels, it was actually too dangerous to go inside. So this cave has a very steep vertical drop almost immediately, so we couldn't physically enter those roosts. And in those cases, we either draped them inside or just put them at the entrance. All other roosts, though, we put them quite far into the roosts where the bats were. We sampled most of them for 24 hours, with the exception of a few. So the tree roosts were in the Archaeological Reserve, and that's open to the public. So we were asked to only survey during closed hours so that the public didn't see us going in there and effectively vacuuming the sky. And they also, because they couldn't go inside the roost, they weren't protected from the rain. So we only ran those for about 6 hours because we didn't want our batteries and our electronics to get destroyed if we couldn't get to them in time. It worked remarkably well. So here's a list of our roosts. and the bat species we detected, and then the other vertebrates. So we detected 11 bat species, 10 of which had either been confirmed in those roosts or the area around those roosts that trip or on previous field trips. We detected several non-bat vertebrates and a couple amphibians, non-vertebrate mammals and then a couple amphibians. And this was really encouraging. So what this means, or we think it means, it's still kind of early days, is that we can semi-remotely and pretty non-invasively survey not just regular roosts, but hard-to-reach roosts with high accuracy. And that can be game-changing for two reasons. So the first is you don't need physical access to the organisms. So this cuts down roost disturbance because you're not having to handle the bat. And that Hello? Oh, let's not on this side now. Okay. Where was I? Oh, yes. So it can reduce disease transfer, either between humans and bats and humans, or between bats themselves, because you don't actually have to handle the bats to figure out what species are in there. And so that can be really important for things like white nose syndrome, where you don't want to necessarily be transferring one disease from one bat to another. It also could potentially reduce the time and manpower needed to survey multiple roosts. So you can set up your samplers in one roost, leave see here, you've got frequency and then amplitude. And you see two little spikes in frequency and amplitude, and that's the big-eared climbing rat. But what you don't see is an elevated background frequency or amplitude. So what we think that means is our samplers are relatively quiet and they don't emit high-frequency noises, which when you're working with a species that is sensitive to things like that, it makes a huge difference if you want that method to be non-invasive. With all those great things, we still have a lot of questions to answer. So we don't know how many samplers we actually need to survey a given airspace to totally get the diversity in it. We don't know how long we need to sample for. We don't know how far it travels. So in aquatic systems, we know that eDNA drifts, and we suspect that it also drifts on the air. And that's important if you're looking at roosts that are close to each other. You don't want to get instances where eDNA from one roost is drifting into your second roost and confusing who's where. So we don't actually know how far it travels. We think it settles out relatively quickly, but again, something we need to know. We don't know if the size of the roost matters. It likely does, but in an enclosed space, we don't really know. And then, as I mentioned before, we don't really know when we need to sample. The goal, really, and the true test of this, will be to develop a comprehensive protocol that would allow for semi-automated sampling and monitoring for the terrestrial ecosystem, similar to what we see in aquatic eDNA, where for aquatic surveys, eDNA is pretty commercially and by the government as well employed to do biomonitoring and biosurveying. And that's something that we would like to replicate with the air, as well as expanding it so that we can ask more ecologically complex questions rather than just, is this species here or not? So that might be things like occupancy modeling. But we've had a lot of interest in this, and so we're really excited to see where it goes. I'd like to give a quick special thanks to all the co-authors who helped me with this paper that will be published soon for York University and the NSERC for funding, and of course all the people who were on the field trip that helped with bat captures, and the Lamanai Field Research Center for all of their help and support.
What an awkward microphone to pass back and forth. We do have time for questions, if there are any. Pardon? Is it working now?
We think so.
Ooh, excellent. Fail.
Go ahead.
This is all news to me, so forgive me if this is a silly question, but, so I'll say this, cool talk, and very interesting. What is the, chance of a false positive? Is there a chance of a false positive?
Yeah.
Specifically thinking that you have two different species, but you actually only have one, for example.
Okay, so the question was, what are the chances of a false positive, and the instances of where you think you have two species when you actually have one. So there are false positives are always a concern when working with eDNA, but we have sort of built-in checks and balances along the way, PCR controls, extraction controls, things like that, that then help us when we get to the bioinformatics stage to filter things out. So we had the added benefit in this field site of having a very specific and well-documented historical set of data for what was in the area, which of course isn't going to always be the case if you wanted to deploy this kind of fresh. So then you're looking at things like, if you look at your PCR positive control, if it's contaminated with something or your negative control, sorry, if it's contaminated with something, which when you're working with vertebrates is usually you look at the read count, which is the amount of DNA in it, and you typically get rid of any hit or detection that has a read count lower than that. You do have to be careful with that because sometimes, again, you can get rid of true positives that way. That's the very, very conservative way of doing it. There's other ways that are a little bit more forgiving. But you can also look at range maps and stuff. So in one of our other experiments that we did in that field lab, we were getting detections of bats from Africa and Europe and stuff, and we're like, that's what? Turns out people had worked with those species before coming, and we think what was happening is we were getting secondary transfer on their gear. So it is very sensitive. So yeah, those bioinformatic checks are really important, and then replicates. So the white-winged vampire bat, we're pretty sure it's a true positive because it had very high read counts, and we sampled that roost 3 different times, and it was the main hit every single time. Does that answer your question? Okay.
So I guess I think eDNA is cool, but my I guess question every time I hear an eDNA talk is like it's basically presence absence, right? Which is important.
Yeah.
But if you want to know like how many white-winged vampires are there, that's like a challenge. And I know there are ways to get around it. So I'm like with regular eDNA, not Airborne. So I'm curious, like, if you have any thoughts on that?
Yeah, that's. Oh yeah, the question is, so eDNA is often presence absent data. Can you get the number of individuals, correct? Yeah. That's a really big debated question in the eDNA community, and everybody really wants the amount of DNA to mean something. And there is evidence that it can, but so in that the other study, we did look at that. So for us, there was a positive correlation between the number of bats in the room and the number of detections we got, but there were 3 outliers, ones with really high read counts compared to what they should have been with their abundance, and then one with really low. Those were species, the ones with really high were species that were very hard to tell apart. So my supervisor was basically taking them out right next to our samplers and looking really closely at their teeth and handling them a lot to figure out which was which. So they probably shed more DNA. Whereas the one that was really low was the common vampire bat, and you can just poke the bag and it hisses at you. You don't even need to take it out of the bag to know that it's a vampire bat. So there is some evidence of it, but I think at least for airborne eDNA, it's still too early to tell if we'd actually be able to do that.
Do you have a sense of how long the DNA signature persists?
So the question was, do we have a sense of how long the DNA signature persists? There was a study done out of Texas that used the dust collectors, and it looked at 2-week intervals. So we know that it isn't, in most cases, isn't longer than 2 weeks. And again, that classroom field lab experiment that I was talking about earlier, we noticed that it was a couple of days. So right now, again, that's something that we want to find out, do a little bit more work specifically focusing on that. But for now, it's anywhere between 2 weeks and like 3 days. We think it settles relatively quickly, though. But yeah. Okay, thank you.